major applications that we're taking a look at here, some we're already familiar with. So for example, cancer and molecular biology. Those of you who are doing this work, you'll be familiar with our offering here, being able to measure tumor volumes, perfusion with microbubble nonlinear contrast mode, hypoxia with the oxy-hemo mode on the photoacoustic imaging system, and using this to maybe do tumor model characterization or response to treatment or that kind of thing. There's also a lot of potential in the molecular biology as well, so being able to characterize things like optical dyes and nanoparticles, other photoacoustic contrast agents, and do sort of pharmacokinetic analysis, looking at drug delivery, microdistribution, looking at presence of biomarkers and that kind of thing. So this is something that could be done and is frankly being done with the Vivo laser right now. And I want to just present a few examples. There's this isn't actually a photo of one of our images. This is a histology image, but it comes from a study, and this study was featured. They got a feature on the front of Cancer Research, which is really nice. But this is a study of from one of our customers, Stuart Foster's lab over at Sunnybrook Research Institute, where they actually use the oxygen saturation measurement capabilities of the Vivo laser in this work. There's another one from Scientific Reports looking at, again, looking at oxygen saturation in response to radiation therapy. With the molecular imaging, there's a lot of work going on in photoacoustics right now with photoacoustic contrast agents, and in fact multimodal contrast agents, so something that's visible with photoacoustics as well as with iron oxide MRI, PET tracers, all sorts of different things. So this is very exciting as well. And then here's a review from Dr. Willman's lab at Stanford, Photoacoustic Imaging and Oncology. It's a general review of how this is being used in cancer currently. And here's a really nice paper looking at bladder tumor models and using some of the capabilities like volume measurements and oxygen saturation measurements with the Vivo laser system. So really exciting, lots of nice work going on. I do want to take a look at this one. Just came out very recently. This is a paper out of China looking at oxygen saturation, but this is a really neat thing where they've actually taken nanoperfluorocarbon, which they can then load with oxygen. When it goes to the lungs, it essentially gets loaded with oxygen. It then goes back to the tumor, and by using just a standard ultrasound transducer, you can actually cause that oxygen to be released from the perfluorocarbon. The perfluorocarbon then goes back through the circulation, gets back into the lungs, picks up more oxygen. So this is essentially a really neat way of delivering oxygen in a very specific place. Instead of just using hemoglobin to let it do its job, they've introduced this perfluorocarbon agent, and then they can induce it to release the oxygen wherever they want it. In this case, in a tumor, where they want it to respond better to therapy, the presence of oxygen for things like photodynamic therapy and radiation therapy is very important, so they want to enhance that oxygen in that tumor. And they do that just using typical ultrasound, and they measure the extent to which they've increased the oxygen using the Vivo laser system, the oxygen saturation measurements. So really neat study here as well. This was in Nano Letters. I think it just came out last month, maybe even this month. I do want to point out some proof of concept work that we did here, and I've tried to summarize it in this slide here, where you see we're using sort of iceberg metaphor, where I want to outline and highlight the advantages of photoacoustic imaging and ultrasound in general, just to get from something like this, where a lot of people in cancer research are probably used to looking at something like this, where you have a fluorophore of some kind, and you're able to identify where approximately that fluorophore is in a whole body photograph. But if you want to go deeper, that's just the surface. If you want to go deeper, you really need a different modality in order to do that. And photoacoustics really allows you to do that. This is a sub-Q tumor, but it's, again, a very small tumor. It's about 2 millimeters, 2.5 millimeters in diameter, but you're able to see with, a lot, really high resolution very clearly here. And I'll sort of describe what these things are in the next slide. But, we want to go from here, the surface, deeper down. We want to see things like oxygen saturation, blood flow, tumor volumes, and that kind of thing. And this was sort of a case study that we looked at, some proof of concept work that we did in collaboration with a company called Optimios, who have licensed a technology from Princeton University. It's a nanoparticle technology that we're able to detect with our photoacoustic imaging system. And we address some of these questions that remain after just pure optical imaging. Are we getting specific versus non-specific uptake here? What does the microdistribution look like? How deep is it? Where are the necrotic areas? Looking at real-time pharmacokinetics, how quickly are these things coming out of the circulation? So we've answered a few of those questions using our system and using these nanoparticles from Optimios. I do want to say, though, that was all done on the Vivo Laser, and I think with the Vivo Laser X, what are we really looking at here with a new system? We're getting extra speed, faster image acquisition, and better depth and sensitivity, and that can really be applied to something like this, where, with any imaging modality, using contrast agents, people are always looking for higher sensitivity, more depth, and better resolution as well. There's another company who makes nanoparticle contrast agents. They make gold nanoparticles, and that's Nanohybrids. And they actually spun out of a lab at UT Austin, Stanislav Milyanov's lab, where they published this study. So this is a really nice study looking at metastasis detection, where they established a tumor in the tongue of an animal. This is the ventral surface of a mouse, and there's the mouth and the nose up here. They established a primary tumor in the tongue, which then metastasized down through the lymphatics into the cervical lymph node. Sorry, I'm just getting... We're back. So they established this this model, which metastasized down through the lymphatics into the cervical lymph node, a lymph node in the neck. They used optical imaging to be able to detect the metastasis, but when you really want to look at these micrometastatic sites and see where you're getting this seeding, they used nanoparticles which were targeted to these tumors and which looked optically different if they were taken up by tumor cells. So if there's no tumor, you didn't see any signal from the nanoparticle, whereas if you had this targeted particle and you had a metastatic site, you'd be able to see it here. So really nice study. So I do want to point out, we do have these partners that if you are interested in doing any kind of contrast imaging with photoacoustics, we welcome you to contact us or contact these companies to talk about what they have to offer. A lot of the contrast agents that they make can be functionalized, can be customized, so a lot of interesting possibilities for molecular imaging.